Yes, we can start. Okay, thank you. Okay, good morning and good afternoon to everyone. Welcome to another Global Science Exchange Series webinars of ASCE, focusing on environmental issues, discussion by early career researchers. We have four young researchers. And today the topic of the webinar is the wastewater-based epidemiology studies, which received great attention during the pandemic. First of all, I would like to introduce myself. My name is Bilge Alpaslan Kocamemi. I am Associate Professor at Marmara University Environmental Engineering Department in Istanbul, Turkey. I am a Principal Investigator and Scientific Advisor of Turkey in nationwide tracking of COVID-19 by Wastewater-Based Epidemiology project. This project is ongoing since the start of the pandemic, more than 2 years. Therefore, as being a scientist heavily focusing on wastewater-based epidemiology studies more than 2 years, I'm really excited to moderate this webinar of ASCE on wastewater-based epidemiology. I'm glad to be here. Yes, as you all know, with the start of the pandemic in April 2020, countries have started to monitor COVID-19 distribution in their community by scanning wastewater. And as of today, according to information from COVID-19 Plus dashboard of University of Merced, maybe Colleen is here, I'm not sure. Thanks to Colleen and her team for this dashboard. It's a great dashboard. And today I checked from this dashboard, and for now, 67 countries in the world performing the wastewater-based epidemiology for COVID-19 tracking. And hopefully, in all over the world, the COVID-19 case numbers have started to decrease, and the pandemic will be over in the very near future, hopefully. But with the experiences gained through wastewater-based epidemiology studies, we are ready for early detection of COVID-19 waves and for new pandemics. But more importantly, the sustainable use of wastewater-based epidemiology in the post-COVID-19 period will accelerate One Health approach by providing us an opportunity for systematic tracking of viruses, other viruses, pathogenic organisms, antibiotic resistance genes, and most importantly, the other contaminants of emerging concern. However, these are the good news, but however, there are still many challenges of wastewater-based epidemiology studies need further improvement. And one of these challenges is the inadequate communication between public health sector and the water sector and the lack of modeling. And today, our first speaker, Hoffman, who is the postdoctoral research fellow from Harvard School of Public Health, yes, will talk about the fecal shedding models for SARS-CoV-2 RNA. I would like to give words to Till. Yes, the floor is yours, Till.
Great. So coming through all right?
Yes. Perfect. Please go ahead. Okay.
So yeah, we'll be chatting a little bit about fecal shedding, and that's interesting because of course we need to know what's coming in to be able to understand what we're taking out at the other end in the treatment work. So just as a brief reminder, suppose RNA is shed in feces, then what can we do with that? Treatment works, of course, aggregate wastewater from all sorts of people, and that means we can get relatively cheap surveillance of the infection in the community, because we only need to take one sample for all the people that are within the catchment area of the treatment work. It's also appealing because everyone goes to the toilet, so it's very representative. We don't have the issues that we sometimes see in clinical surveillance, where people are skeptical of the healthcare sector, and we have differential testing in different demographic groups. But having said that, we of course need to understand how much RNA is shed in feces, which I'll be talking about. But there's a number of other issues, such as degradation of RNA within the transport, and sewage is of course nasty stuff. So it's sometimes very difficult to analyze samples as well as we would like. Sampling strategy is also important. You know, where do we take these samples? How often do we do it? But I'm sure we'll learn more about that in the other talks. So what might we want to know about in the context of wastewater-based epidemiology? Because there are a number of other questions that could be of interest. So, for example, how does RNA end up in the stool? Is there some sort of gastrointestinal infection, or is it just that people swallow their own spit that's got RNA in? Can shedding tell us anything about the disease cause? And finally, is there a risk of fecal oral transmission? But remember, detecting RNA doesn't mean that there's live virus. So what might we want to know? One is the shedding profile. So over time, what does the amount of RNA look like? That might be interesting because, for example, if we have a very sharp peak and then a quick decay, we'll see a different signal in the wastewater as opposed to if we have a sort of rolling profile where it starts slowly and then decays slowly. And then, does everyone shed? Is every infection leading to fecal RNA shedding? And again, that might be interesting because we might have to adjust our prevalence estimates, so the number of people in the community that are infected, if not everyone does shed. And then, of course, how many RNA copies are typically shed, because that allows us to do a prevalence estimation, or in the wastewater community, it's also often called back calculation. And then finally, what does that mean for the samples we might analyze? So quick PCR reminder, we get a sample that might be stool or wastewater. We make lots and lots of copies of the RNA using PCR, and then we measure how many copies we've got after making lots of them. So one issue is that there is a level of detection or level of quantification. And if the concentration is below that level in the initial sample, we might struggle to get a meaningful signal. And that's an important thing to keep in mind. So what are the data we might have access to? So we've got a number of different patients, and from each patient, we'll collect different samples. And I've sort of illustrated quantified samples with a little blue circle here, and then we might have negative samples. And negative here means below the limit of detection. And these patients might be members of different studies, and the protocols will differ slightly between studies. We might have different levels of detection in different studies, so the data is a bit of a mess, really. So what can we do with that? So here is a conceptual illustration of a model to try and get as much out of these data as we can, given that they're not the nicest data that one could imagine. So digging into this a little bit more, let's first have a look at one patient. So we'll have the solid curve that we can see, and that represents the infection course of one particular patient. And illustrated in orange here are samples that we've taken from that particular patient. And we've got a few samples that have been quantified, so we really know how many copies there are per milliliter of stool. But we've got this dashed line that tells us the limit of quantification, and samples below that level we'll consider negative because we just don't know how much it is in there, except we know it's less than the limit of quantification. So that's the profile for one patient, and the sort of shaded regions illustrate the variability within that particular patient. And then we've got a number of different lines here that are dotted, and that is meant to illustrate the variability between patients, because someone might have a much more intense infection. Maybe that leads to more RNA shedding. And the sort of blue distribution here illustrates that variability amongst the whole population. And something that is also shown here, so early shedding, we currently don't have much data for, because all of these patients for which we do have data are in hospital. So it's been at least a couple of days that they've been ill, and we really don't know much about the beginning. And yeah, people don't like collecting their own poo in the first place, and they particularly don't like collecting it when they're feeling quite ill. So that is an open problem in terms of understanding the data better. So that was the concept. Here's the reality. I've plotted in different color samples taken from different studies, from different patients. And the horizontal lines are the limits of quantification in different studies. And you'll notice that there's almost 2 orders of magnitude in differences in terms of limit of quantification. So different studies can really tell us very different things. And we have here a fit, and admittedly, it is not the best fit that people have ever seen, but we are relatively confident about it. That shows us the sort of decay profile. And this is, again, the late stage decay profile. And the half-life of that profile is about 1.5 days. So what does that tell us in terms of the shedding profile? Yeah, it's decaying, as we might expect, as people are recovering with this 36-hour half-life. So things are really decaying quite quickly. And this is nice for us because wastewater-based epidemiology really might be able to provide an early indicator for infection that's happening within the community. Because typically we'll have to wait until people feel a bit rough, and then they get a test, and then we analyze the test, and then we have some data about what's going on. But if most of the shedding happens early on, before people actually feel ill, then we might be able to have a leading indicator of infection from wastewater-based epidemiology. But you might say, you know, you've just picked an exponential profile. What would have happened if you'd done something else? So we did exactly that. We tried some different shedding profiles. So on the left here, we've got gamma distributions as shedding, gamma probability distributions as shedding profiles. On the right, we've got a shedding profile proposed by Toynis that was from norovirus shedding. And what we see here is all these little blue curves are possible shedding profiles that are consistent with the data that we've got observed. And you'll notice that in the late stage, sort of in the white region here, as opposed to the gray region, we really can constrain the shedding profile quite well. And it doesn't really matter which shedding profile we take. They all look very similar to an exponential. But in the region where we don't have much data, all sorts of shedding profiles are consistent with the data. So for example, on the left, the shedding profile might peak around sort of, 10 to the 6 copies or around 10 to the 9 copies. So we really don't know very much. And that's not surprising because we just don't have any data. So the location and or the magnitude of the shedding peak really can't be constrained at the moment. And there might be new data that's come out since we've written this paper. But any results that sort of say the shedding peak is exactly this amount and at this time, that's likely a modeling artifact, or there's new data available that we haven't heard about yet. So does every infection lead to fecal RNA shedding? Well, that depends on what we mean by does someone shed? So one might say that if someone sheds 0 copies, then they don't shed. If someone sheds 10 to the 9 copies, they do shed. But where do we draw that line? And in practice, we often draw that line at the level of detection or quantification of the assay that we're using to analyze the data. But that again depends on the method we're using. And from a scientific perspective, that's not particularly appealing because ideally the answer to the scientific question isn't severely method dependent. So the approach we took here is we've introduced a variable P, which is the probability that each patient sheds, as in the model that we looked at before. And with probability 1 minus P, they do not shed any RNA at all. So in the studies, if we have a patient who has even 1 positive sample, we know they do not fall into the category of the non-shedders. And only people who have never had any negative sample could possibly be in this group of non-shedders. So if we analyze the data, we get an estimate that the best estimate of the proportion of people who shed is 0.97, but the 95% credible interval is somewhere between about 80% and everyone shedding. So we don't really see much evidence for the subpopulation of people who do not shed RNA. And there have been a number of studies who've sort of said it's around 50% or so. But of course, again, having a negative sample doesn't mean that there isn't any RNA, it just means that there's less RNA than the limit of quantification. So again, how many RNA copies do we typically see? Again, that depends on what we mean by typical. And I'm going to argue that for wastewater-based epidemiology, the only thing that's relevant is the mean. Because when everything mixes together and we take a sample out, then effectively we see the average of all the people who've been in the catchment area. And recall that the mean on the log scale is not useful because the mean of the log is not the log of the mean. So we really need to model this carefully. But again, we don't know very much about early shedding, so estimating an average is tricky. So what we're going to do is we're going to say there is actually no shedding profile. We're just going to assume that there's constant shedding over the disease course. And if we apply that to the data from these hospitalized patients, we get a number of about 2 million copies per milliliter. But notice that the credible interval is about 3 orders of magnitude. And that is, of course, not very nice, but that's because we don't have very much data. And means are very, very sensitive to outliers. So if we had looked at the median, for example, the median number of copies, we would have had much better confidence intervals. But what we care about is the mean. We've also asked here the patient level distribution to capture all variability. So we've said there is no shedding profile. So both early stage samples or relatively early stage in the hospitalized setting and late stage sample need to be captured by the same distribution. And that leads to more variability and also to these terrible credible intervals. So what should we expect when we analyze wastewater? So the number of copies we'd like to, or we expect to see in wastewater is the concentration of RNA in feces times the feces volume divided by roughly the per capita wastewater volume that a person generates. So we've used some values from the literature, about 130 grams of stool per day per person, and about 300 liters of wastewater per person per day. That will, of course, depend a lot on people's diets, culture around water use, etc. But that spits out roughly 10 to the 3 copies per milliliter of wastewater. But remember, this is if everyone was infected, because we've just looked at behavior of one person assuming that they were infected. So in practice, that number is going to be lower. So we'd get about 10 copies or so if 1% of people were infected. In practice, we see higher numbers. And that lends further support to this hypothesis that the shedding peaks early, and that we've really only seen a small part of the RNA copies being shed amongst these people who are in hospital. And again, this early stage shedding is outside the observation window. So in light of that, what do we know more about? Shedding profile, about 36 hours half-life in the late stage, but we really don't know what's going on in the early stage, and we need more data for that. Does every infection lead to shedding? Probably yes, because the number of negative samples we see are consistent with the level of quantification in these studies. Then how many RNA copies do we typically see? Around 1 million, few factors here and there per milliliter of stool. And then what should we expect to see in wastewater? On the order of tens of copies at 1% prevalence, according to the analysis here. But in light of the shedding profiles we've seen, we'd probably expect to see more. Even though, I've had lots of answers that were, well, that depends, or the credible intervals are large, we're still quite confident in the modeling that we've done, because we've done extensive validation checks, including making predictions for studies that were not included in our model fitting and seeing whether our predictions about those studies were consistent with what people actually observed, and they indeed were. So if you want to find out more, unfortunately only a preprint at the moment. We've been battling the long review process in the COVID pandemic, but hopefully we'll have it out as a proper paper soon as well. And that's everything from me.
Okay, thank you, Till, for this great presentation. I'm just checking for the questions through the chat box. As far as I see, there is no question now. I may ask one question while we are waiting for the other questions. What do you think about the following, the scanning the monkeypox? You know, especially in Europe, we are discussing, can we scan the monkeypox in the community through the wastewater-based epidemiology studies? And as far as I learned from the veterinary doctors, there is not much data on it. What is your thought about this?
I also have to say I don't know very much about it. I haven't heard yet whether there is fecal shedding in monkeypox infections.
Okay. Yeah.
Sorry for this. I didn't understand what he's talking about. So, Till, I may ask one more question. You show the graph showing the data for early shedding and you know, the after the symptoms appear. So certainly I'm just looking to those graphs in the wastewater-based epidemiology side, and we are always emphasizing that the wastewater-based epidemiology studies are quite important, especially for the asymptomatic patients. And as far as I know, I'm also in contact with the medical doctors in our country as well. As far as I know, there is lack of data about the asymptomatic patients. Do you have such data, and what do you consider? How can it be planned because it is asymptomatic, and how can we obtain such data? Because it will be necessary for the wastewater-based epidemiology modeling studies in future. So what do you think about this? Phil, can you hear me?
Yeah, sorry, I had a bit of a glitch. I think I got the question. So
I can repeat if you want very briefly. I'm just asking the lack of data about the early shedding. How can we collect data from the asymptomatic patients? Because this data will be necessary for the models. So what do you think about this?
So we've thought about two approaches. One is, for example, in the UK, there is testing for colorectal cancer, and people are sent little test tubes that they send back to their doctors to test for cancer markers. And we might also run PCR tests on those for quantification. But of course, that's only useful if the level of prevalence in the community is relatively high, because otherwise we need to run so many tests before we get a few positives. Another one is that some universities are running challenge studies where they actively infect people with SARS-CoV-2, and we might be able to get information there, because typically those are people that wouldn't be captured in cohort studies in hospitals, because they tend to be younger. They want to make sure that as part of the challenge studies, people don't actually, get severely sick. So they take relatively healthy young volunteers. And that would really be quite complementary data to what we have at the moment for people who tend to be older in hospital at the late stage. But it's very difficult from an ethical perspective.
You said young people. Do you consider the age, gender, the ethic, those are factors affecting the shedding concentration?
Again, one of those questions where I have to hold up my hands.
I'm asking difficult questions to you. I know.
Okay. I think probably in sort of respiratory shedding, that might be something that we can analyze, because we've got millions of samples. And there's enough data to be able to sort of drill into, is there a demographic component to shedding? But I think in fecal shedding, it's already so difficult, even if we pool all the data, that it would be very difficult to make any sort of demographic distinctions, I think. But of course, if there's more data, that'd be great.
Okay. We have questions. Alamin is asking, how many days ago early warning in sewer shed? This is quite important. Can you answer this question? Roughly, how many days ago early warning in sewer shed? I can answer, but I would like to hear from you.
Let's see whether we agree. So from this work, it's difficult to tell. We did some correlative analysis between wastewater samples, and in the UK, there's a COVID infection survey. So this is just a household survey where people get swabbed independent of whether they currently have symptoms to have a prevalence estimate. And there's also, of course, just clinical surveillance. And we did a correlative study there, and there is no lead against the COVID infection survey, as we might expect. But there is about a 3 or 4 day lead against the clinical testing, where people feel ill.
Okay, I can give an example from Istanbul. We are catching the increase in the number of the cases 3 to 4 days, sometimes 1 week before the announcement of the case number increase in the city. And there is another question. Do you see any difference in shedding between vaccinated or non-vaccinated people?
Again, great question. There is some difference in shedding, again, in respiratory samples, but we don't currently have any fecal shedding samples for people who are vaccinated, in part because this work was done relatively early on. But also, people luckily with vaccinations don't get as ill, so there's very few of them relatively in hospital.
Okay, thank you, Till. Maybe we can continue to questions in the last part. As you know, the sustainable use of wastewater-based epidemiology in the post-COVID-19 period requires private sector participation to those activities. And our next speakers, Kyah and Denise Prussen, are from a well-known global privately owned firm, CDM Smith, providing smart solutions in water and environment. Both Kyah and Denise are environmental engineers, and they will talk about the experiences and lessons learned from applied wastewater epidemiology. Yes, go ahead, floor is yours.
Thank you. Yep, I'm gonna go ahead and share my screen now. Can someone confirm they can see it?
Yep, I can see it.
Great, thanks Denise. All right, so hey everyone, as was just mentioned, my name is Kyah, and together with Denise, we're going to be talking about experiences and lessons learned from applied wastewater-based epidemiology, and in the form specifically of a wastewater surveillance program. So a quick overview of the topics we'll be covering today. We'll talk about what is wastewater surveillance, why you would want to implement a surveillance program like this, specifically the civil engineer's role in a program like that, and then we'll also talk about the efficacy of composite versus grab samples and some public health actions that can result from wastewater surveillance, and then also touch on some future uses of this kind of work. So as everyone knows at this point, wastewater surveillance and wastewater-based epidemiology has historically been used to track viral diseases as well as illegal drug usage in communities. And as we now know, people shed inactivated SARS-CoV-2 virus in their bodily fluids, which can be used kind of as a fingerprint and measured in wastewater. Now, the real power from this, as we were just discussing in the Q&A section, is the ability to detect the presence of the virus up to 7 days before people feel sick enough to get tested. So this has the potential to give public health professionals up to a week of additional time to take action to prevent the virus from spreading, because it gives those results potentially before clinical testing, which is huge when in the context of outbreaks during a pandemic when time is really of the essence. So looking back to prior to March 2020, techniques did exist for analyzing viral diseases in water, but wastewater surveillance itself was not really widely used, and programs existed in less than 10 countries. Now, also in March 2020, both the academic and consulting engineering communities were looking for ways to be of service when it came to the COVID-19 pandemic. And as I mentioned, we knew at the time that people shed the virus in their bodily fluids and that academic work had been able to quantify traces of SARS-CoV-2 in wastewater using new techniques. At the time, you may remember we had severe shortages of PPE, limited testing, and we didn't really have real-time information on who was sick or how to properly protect ourselves. So when early on it was discovered that it could be possible to detect the virus up to 7 days before clinical testing results, it really accelerated and increased the application of wastewater surveillance as a way of measuring these pathogens in wastewater. And in turn, it's really advanced the use of wastewater as a source of information. So as you can see in this graphic, now jumping ahead to March 2022, there's wastewater surveillance programs in over 64 countries, some at universities with over 3,000 sites. And we began our work with wastewater surveillance for SARS-CoV-2 in March of 2020. So again, looking back to early 2020, utilities and public health departments were not really used to communicating with each other. And in order to have a successful wastewater surveillance program, we as the engineers had to bridge that gap between these entities. So the first step with a program like this would be to engage with health agencies to establish an approach and objectives, which could include identifying appropriate and possibly vulnerable populations to monitor. So as engineers, we brought in our knowledge of collection systems and our ability to provide field staff for the program, which allowed us to select ideal sampling sites and mobilize to collect representative samples. So when planning a wastewater surveillance program, scale really matters. For instance, if you sample at a treatment plant, you're getting an entire city's worth of data. And on the other end of the spectrum, if you sample at a single house, you're not really getting a representative sample of a community. There's kind of a sweet spot in the middle in terms of scale. And as engineers, we were able to pinpoint and refine the proper application of wastewater surveillance by adjusting the scale and usually looking on the more neighborhood and congregate care facility level. So steps 4, 5, and 6 seen here all kind of deal with analyzing data and working to get the data into the hands of public health officials who can take action. So here we look at ways to get the data as quickly as possible and ways to refine analysis techniques and reduce turnaround times for data analysis. And in a bit, Denise will actually talk more about that part of our program. So why would you want to implement a wastewater surveillance program? We're going to look at some key motivators and advantages and results it can provide. So a huge one, pardon me, is providing early warning for new outbreaks. So this graphic on the right is from a study done, I believe, at Yale, and it shows that one of the earliest assessments of that lead time for wastewater surveillance compared with clinical sampling. So the other advantage is you can capture asymptomatic and symptomatic individuals. So people, as we know, shed the virus, whether they're symptomatic or not. And someone who's asymptomatic might not think to go get tested, but again, wastewater doesn't lie. And then also it's not dependent on healthcare access or voluntary clinical testing. So it's easier, it can be easier and more cost effective than the nasal swab techniques that have been used. So a few more advantages or reasons you might want to implement a program would be you can detect variants, you can confirm the absence of the disease, confirm clinical trends you might be seeing. It's an anonymously pooled sample. And then also it's easier to monitor larger populations than clinical testing, which in turn leads to a lesser burden on public health departments. So, and then also looking at the future and present here as hopefully cases die down and we're also seeing home testing ramp up, there's now potential for a lack of data and reporting if, people are just testing at home and not reporting those numbers. But like I said, wastewater testing doesn't lie, and you can still, with wastewater surveillance, get an anonymous measure of how much COVID you might have in a community. So looking at the role of a civil engineer in a wastewater surveillance program, again, as I mentioned, we have the knowledge of collection systems, the ability to mobilize quickly, and then there's a lot of troubleshooting that we conducted in the field as we were rolling out these programs. So as an example, one of our sampling sites was a congregate care facility, and the first sampling location, the first manhole we selected, proved to have a lot of issues, particularly with viscosity, to the point where the labs we were sending it to, their quality controls were failing, and we were actually unable to get proper data. So here we had to take into consideration if there were low flows, potential sediments getting in, industrial additives maybe added to the wastewater. We looked at manhole flow dynamics and all of these things and used our knowledge of the system to identify a manhole that was further downstream, but that would still give us a representative sample just for that facility. So we used dye testing to confirm that we had a viable site and ended up a bit downhill from our original site, and we were able to get good data after doing some troubleshooting. So we implemented this wastewater monitoring at schools, correctional facilities, congregate care facilities, as I mentioned, and then on the neighborhood level as well. So as we conducted wastewater surveillance through these various locations and programs, we were working to standardize the techniques and protocols for the sampling. Now, when determining effective sampling locations, we would consider at the amount of flow, if we could get a representative sample, and if even the location or manhole could even handle the installation of an auto sampler. So the physical variability also of wastewater made each sampling site have different characteristics to work around. So some locations, the concentration levels might have been affected by other sanitary flows, maybe like from rainfall or other infiltration or even industrial wastewater getting mixed in. And then time of day can also cause variability in wastewater characteristics. So the time of day consideration led us to sample using an auto sampler. So on the right over here is an example of the auto sampler we used, and this is it hanging inside a manhole with a sampling tube. So this allowed us to get a time composite sample. So they would be programmed to take a sample every 15 minutes over a 24-hour period. And this allowed for the time variability in the flow to have less of an effect on our sample and allowed us to get a more representative sample. Now, there were times when we had issues with the auto sampler not giving us a sample. And in this case, we would take a grab sample, which is a sample taken at one point in time on a given day. And when we started doing this, we considered that taking grab samples would be easier and actually cost less if we could use it as our main sampling method. And so we wanted to see if a grab sample could provide comparable results to a time composite sample. And Denise is actually going to talk more about this now.
So as Kyah just mentioned, one of the main motivators behind our research and development lab analysis was, is there a drastic difference in values of SARS-CoV-2 obtained in grab samples versus composite samples? And our other main motivator was, can we detect SARS-CoV-2 in wastewater through our internal own in-house lab analysis? So going based off the first point, collecting a representative sample. So as Kyah just mentioned, typically we wanted to collect a composite sample at these manholes. So the auto sampler would collect an aliquot every 15 minutes over that 24-hour period. But then when, for some reason, a composite sample could not be collected, like Kyah just mentioned, due to viscosity of the sample or some sort of clogging in the tube, our sampling team would then take a grab sample through just the use of like a dipper, dipping down into the manhole and collecting whatever water is currently in that manhole. And that just captures a specific moment in time. So we conducted some lab analysis to quantify the difference in SARS-CoV-2 when a grab sample is collected at the same time that final composite sample is removed from the manhole. So overall, our results show that there's a really low correlation between grab versus composite samples. As you can see on the graph on the right, we put an R equals 1 line there just to show the comparison that there's very low correlation. And one very crucial point is that in many cases, our grab samples would show no virus and our composite samples would be showing virus numbers. And this is primarily due to the fact that a composite sample, you get more of a chance of collecting the stool and the wastewater of someone that is infected, versus a grab sample, you could just be collecting someone that's currently taking a shower at the time or washing their hands or running a washing machine. So a composite sample is, as proven here, a composite sample is a much more representative sample as to what's actually, as the numbers of infected people in a community. So our other main motivator behind our lab analysis is, could we actually analyze samples in-house as a civil engineering company? So why would we analyze samples in-house? So when this idea was originated in March of 2020 originally, we were basing it off the assumption that there would be very few external labs actually performing this analysis, and they would be most likely overwhelmed by the sudden influx of samples at the beginning of this pandemic. In addition, we wanted to look into providing quicker turnaround times for public health departments. So as mentioned a couple of times before, why do quick turnaround times matter? So if there's detection up to potentially 7 days before symptoms, this could prompt our public health departments to take quicker public health actions. And in turn, we're protecting some of our most vulnerable communities, such as the congregate care facilities, public housing, schools. So what specific public health actions can be taken from our sampling? We saw resident notification in buildings, neighborhoods, and schools, mass distribution, and mobile testing deployment. It's really important just to make the community aware of the sudden uptick of cases in that area. So as you can see on the graph on the right, this is just one of our sample sites showing an uptick in cases shortly after the holiday season. So overall, we wanted to try and reduce our turnaround times in order to provide quicker results to our clients, to the public health departments. So we conducted in-house lab analysis. Our in-house lab analysis took a total of approximately 8 hours to complete through qPCR analysis. As you can see in the figure on the right, that is an overall steps that we would follow. We would do sample preparation, concentration, RNA extraction, and RNA measurement through the use of a qPCR machine. So qPCR essentially works by amplifying the DNA in the sample through essentially cycles of heating and cooling. And then the cycle at which the SARS-CoV-2 is expressed can then be translated over to a value of SARS-CoV-2 in copies per liter. So we were able to conduct this analysis in a total of about 8 hours. However, when we sent out samples externally to labs, we would get results in approximately 1 to 3 days. So overall, we were able to prove that we could provide results to public health departments up to 3 days faster. So now looking at wastewater surveillance beyond COVID-19, as we now have seen that wastewater surveillance has been proven to be effective with this COVID-19 pandemic, this can now be used also with other viruses as listed on the right, on the left. And then in addition to other viruses, wastewater-based surveillance could also be used to track antibiotic-resistant bacteria and other healthcare acquired infections. So overall, our lessons learned were how to implement a successful surveillance program, including a lot of troubleshooting in the field, confirming absence or presence of disease, which is especially crucial during times of low prevalence, like now, the specific public health actions that can be prompted from our quick turnaround times, and in addition, the significant difference between taking a grab versus a composite sample. So thank you so much to everyone listed here, and we now welcome the opportunity for any questions.
Bilge hocam. You're muted. Can you try again?
Okay, yes, thank you. I think you just blocked the microphone, so I couldn't open, okay? Sorry. We have questions. First question is why there is a difference between graph and composite samples, SARS-CoV-2 concentrations. I think you answered this question, but maybe you can quickly answer again.
Sure.
Yes, please.
Yeah, sure. So the composite sample would be taking a sample from the manhole every 15 minutes or so over a 24-hour period. So you have a much higher chance of actually catching the bodily fluids and the fecal matter of someone that is infected versus a grab sample. Someone just upstream could just be showering, washing their hands or or maybe running a washing machine. And then those types of activities would be drastically like lowering your chances of catching any sort of RNA of SARS-CoV-2 in the wastewater.
Okay.
Sorry. I was going to expand on that a little bit. Just one more thought. So on the field site level, you're sampling typically, or we were typically sampling at a manhole. So a manhole is just a single point in a collection system. So there's constantly water running through. So if you're doing a grab sample, you're just getting one point in the system at one point in time. So that's where the composite sample can be so much more effective, as Denise said. So I just wanted to point out the concept of the manhole there.
And Kyah and Denise, Till has a question. He asks, did you see any difference when you take a sample upstream, I mean, close to the source, and when you take the sample from the entrance of the wastewater treatment plant and downstream? I mean, do you have such a comparison between the SARS-CoV-2 concentrations, upstream sampling and downstream sampling?
Yeah, I think so. In terms of in-house research that we did, I don't think we did a direct comparison between those two different scales. I think we were more focused on getting the information about specific locations and seeing how those different scales could give public health departments information, and a little bit less focused on necessarily the specific scientific portion of the lab testing. We were more worried about just, are we seeing SARS-CoV-2 and can this information be useful, basically, that we're getting.
Okay, there are the questions related with your lab procedures. I will just wrap up those questions. You said that you are somehow concentrating your sample. Which method you are using? And the Jacques is asking, do you try different pre-concentration methods? And I really wonder which method you are using, because you said that 30 minutes for the pre-concentration methods. And do you have such a comparison between the different pre-concentration methods, different PCR, for example, qPCR or digital PCR? Did you try different methods?
Yeah, so to go based off first the sample concentration question, when this was initially, when we were initially looking into this, we didn't even actually, there weren't even set procedures developed quite yet at the time, but as time went on, then step procedures were developed. But the procedure that we were using for the actual sample concentration was the addition of a few coagulants and then centrifuging the samples. We were using, we were adding certain coagulants, and then after, so our first step was preparing the sample. So first we were putting the sample in a heat water bath to inactivate any sort of active pathogens. And then we were doing sample concentration. And that was just through adding coagulants, pouring out the supernatant, and then centrifuging.
Okay. Yeah. So you just try one pre-concentration method, and I think you are using the RT-qPCR method, huh?
Yes, that is correct.
Okay. I think we have limited time, so I have questions, but I will ask if the time is left at the end of the presentation. And as you know, we have one more question. Maybe we can ask this. Alamin is asking, did you try different samples? I mean, you just monitor, you are just performing the test with the liquid part of the treatment plant, or did you try primary sludge or waste activated sludge?
Yeah, we only did liquid. We didn't try anything with sludge.
Influent, I think, huh? You are just measuring influent, not the effluent.
Yes, that is correct.
Influent. Okay. Thank you. At the end, maybe we can ask a few questions to you. Thank you for your great presentation. As you know, during the COVID-19, we all realized that the viruses evolve, and as they spread among the people over time, the new variants, significantly different than the original virus, are appearing. To identify the new variants, the scientists mapped the genetic material of viruses through genomic analysis. And our last speaker today, David Dreifuss, who is a PhD student in Swiss Institute of Bioinformatics, will talk about the detection and monitoring of SARS-CoV-2 variants with genomic analysis. Yes, the floor is yours, David.
Thank you so much for inviting me to this webinar. So yeah, we at the computational biology group of Professor Nico Berenwinkel have been very interested in the genomics of wastewater-based epidemiology, and we have worked on that in a big collaborative project with EAWAG, which is the Swiss Federal Institute of Water Research. So the groups of Timothy Julian and Christoph Ort, who are the real specialists of the question, of course. And also with groups at EPFL, such as the group of Tamar Cohn, and other more computational and statistical groups, such as the group of Tanja Stadler, have worked on this subject also in this collaboration, which is made also with the Swiss government, the Federal Office of Public Health, who is of course interested in monitoring SARS-CoV-2. So our project came from the genomics came from first this realization that SARS-CoV-2 RNA can be quantified in wastewater, as we know and as you've introduced. It's a realization that has been made simultaneously in many groups around the world. So since the beginning of the pandemic, our colleagues at EAWAG have been measuring SARS-CoV-2 RNAs using qPCR and dPCR. And concentration generally correlates really well with the epidemiological insights. For example, our colleagues at the computational evolution group of Tanja Stadler have been computing the effective reproduction number of SARS-CoV-2 in the population based on this wastewater data, with a very good match also with estimates that can be derived from the number of confirmed cases or death.
David, sorry for the interruption, but we have difficulty with your noise. There is a noise in your, with your voice.
My apologies.
David, if it will help, you can close your video and we can hear you better. I think there's an internet connection.
Okay, you think it's okay? I will just.
Let's try this way. Yes, thank you.
Do you hear me better like that?
Yes.
All right. So we'll continue. One second. I just need to. So yes, from this we have deduced that the next logical step, when the first variants of concern were detected in other countries, and we had the worry that it would be introduced in Switzerland, was to try and look for them in those wastewater samples. So we had the questions lined up like this. First of all, is it possible to even detect them at all? And in that case, if detection is possible, can this detection be made quantitative? Can we quantify the relative abundance of these different variants? And if that is the case, what kind of epidemiological insight can be drawn from this? So our general pipeline is that we draw some 24-hour composite samples from the wastewater treatment plants. We extract, concentrate, and verify those samples, but I'm not a wet lab person, I'm purely computational, so I cannot really comment on that. And then we do some different kinds of genomic analysis, such as multiplex assays of digital PCR or sequencing analysis. We've been doing that in wastewater treatment plants around the country in different areas that cover a good portion of the population and quite well scattered around the country. For almost a year and a half now, maybe a bit more than that. And then we have different types of data. So we have digital PCR duplex assays, where I will not go into the details of the methods here. I think I will focus a bit more on the results, which I think are more telling about the interest that we have in the methodologies themselves or in this type of data. But quickly, in those duplex assays, what we do is that we have some deletion or variable probes that are located closely on the genome to a universal probe. And it's a PCR-based assay, and the patterns of fluorescence that we will see will inform us on the concentration of a certain mutation relative to the concentration of total SARS-CoV-2 RNA. Then we have also next generation sequencing. So for this, we've been using the RT protocol, which is a tiling amplicon protocol. We can recover generally the all of the SARS-CoV-2 genome using 98 primer pairs that will produce amplicons that are overlapping over the entirety of the genomes. And for this, we have developed also some methodology to call the mutations that can co-occur on short regions of the genome to give us more confidence in the detection of this these variants. And this is available as a package as we call Cojac. And this we can compare to clinical data, which can be clinical sequencing, or also qPCR that sometimes have a target failure, and it can inform on the proportion of a mutation. Now, for early detection, using the methodology Cojac that we have detected, that we have developed for we have had sometimes wastewater multiple weeks in advance before we started seeing the introduction of the variant in the clinical sample. So that was for the first variant that we analyzed, alpha, and this signal was confirmed with retrospective sequencing of the faeces. We were pretty confident about the detection. We've repeated things for pretty much every variant that has been introduced on the territory. This same observation, that the first clinical sequence and the signal generally come at the same time. In half of the cases, it came earlier in the wastewater, and sometimes we could even detect the transient introduction of the variant that did not result in community spread, as is sometimes the case that we can detect that with a we are pretty the sensitivity of it, because we're comparing periods of time where a hypo were sent for now it doesn't work only in Prussen. So for example, our collaborators introduction of variants on the British TV, as you have seen, there are briefings that are very detailed. Now, beyond the detection, what about quantification? Are the relative abundance around quantifiable? Depending on the technique, is different. If we're looking at digital PCR duplex assay, like I briefly described before, we're looking only at a single mutation. multiple assays, multiple conditions. We have to select them carefully, so they would be representative of the variant that we are trying to monitor. But then it works very well. This technique is absolute terms, doesn't need a reference curve. It's an application that can be achieved with that. And from sequencing data, since we are looking at the whole genome, we can see the prevalence of a whole set of mutations. And some of these mutations might be shared between. So we have to sort to reconstruct the relative abundance. This is what we would think to do computer packages to do that. Now, from this quantitative, just tracking the what can we do with this epidemiological parameter that important to explain when a new variant is introduced, which relates to its fitness advantage. So, epidemiological models on the waste, those parameters with the same, get the same parameters that we would get, and with similar levels of precision, with orders of be something that for us indicates the edge of wastewater population level estimates. So the same for data generally get wastewater sequencing here. What we conclude, we can achieve early cases at a much lower, and we can quantify the variance using with much less bias stated in the suffer from the age group, and much that.
David, I'm sorry. I think you have a connection problem for the last seconds. I don't know if it's me or.
You still hear me?
And I can now. So I don't know. Maybe it's just my computer. I'm sorry.
Did I?
No, I asked the other audience, Kyah, and she said she cannot hear clearly too.
Okay, I think that, so maybe David, you have a connection problem, but we are trying our best to hear you.
Yeah, I'm sorry, I cannot really. Camera is already off, right?
Okay, no problem. We try to understand from your graph.
Yes, let's continue.
Okay.
Briefly conclude, the detection, quantification, and achieved at a much lower by looking at clinical derived. We are daily wastewater treatment plants, health authorities, and the public. Still in at ETH and thank you very much. I apologize quite well before. I don't know.
Okay. Thank you, David. Although we couldn't hear you very clearly, your results it seems quite attractive. So thank you for the presentation. I'm just checking the questions through the chat box, and I think there is no question. But I can ask one question while we are waiting for the other questions. How many samples you are running? I mean, I'm trying to understand. I mean, the distribution of the base coverages based on the number of the samples, because as we discussed with you before, in other countries, one of them is Turkey, we are facing problems with the sequencing results because we don't have a chance to run many samples. So do you have any suggestions for the countries who are running, I mean, limited number of the samples? And what is your efficiency based on the number of the samples?
So I don't have anything really precise to tell about that, because we would like to make more precise subsampling studies, because we're sampling every day, 24 hours composite samples, and sequencing every day in those locations. So surely we could sequence less than every day. So for example, UKHSA had a strategy of sequencing 3 or 4 samples by week, I think, and but they were not looking at quantifying. Generally, they were looking at detection. So it also depends on what is the goal. If the goal is to detect the presence of a variant, probably fewer samples are needed than if we want to have high precision in the quantification of the relative abundances of this variant. But I assume that it will then also depend on the size of the wastewater treatment plant, or maybe more the size of the catchment area.
Because RNA degradation is increasing when the catchment area, increases. Okay.
But surely we would like to use our data to get a bit more insight on let's say the rarefaction curve, or the diminishing returns of having more of sampling more closely meshed time series.
What is your thought about the future trend of the NGS analysis for the, for not only SARS-CoV-2, for the other viruses and pathogens, because they are quite expensive methods. What do you consider? What is your thought about this?
I mean, we would surely be very interested to investigate. I personally would believe that that it would have a very good place in the toolkit of wastewater-based genomic epidemiology. But then again, we have very good experience with PCR-based methods, especially digital PCR. But there are definitely drawbacks in both directions. Namely, for digital, like PCR-based assays, you need to exactly know which mutations you're looking in advance to design the probes. And whereas if you're sequencing, you can always retrospectively look back and see if something was there.
So you mean for the identification, yes, we have to continue with the NGS analysis, but after the variant is identified, then we can quantify it by using the qPCR primers. This is what we are.
That's a possible strategy. I would assume, yes.
Yes. Okay. I think we don't have any questions specific to you, but I have to ask the technical staff if you have time. We may ask for the old speakers we may ask the audience. Do you have any additional question to the speakers? Do we have time?
So yes, we can use that time instead of our networking event, 10 minutes, we can spend that time for Q&A. So okay. Please feel free to unmute yourself or use the chat box for your questions.
While we are waiting, again, I can ask a question to the CDM people, because since the time is limited, I just jumped my question. So Denise and Kyah, you just talk about, as far as I understand, as a company, you are just analyzing the wastewater samples. Do you have any other service for the community as a private company? I mean, you are scanning the virus routinely and you are preparing the distribution map and announcing for that community. Do you have such activities, or you are just making a lab analysis?
Okay, yeah. So I think a huge part of what we're doing actually is the field work portion is actually a majority of what we offer in terms of services, because as a consulting firm, we have that ability, we have program management experience. And so our ability to develop a program like this and have staff for it and have the resources and be able to help clients manage something like this. And again, as I said before, kind of be that link between the public health departments and the utilities and ability to convey this information. So while we did do some of that lab work to see if we could, change turnaround times, our big focus was on providing actionable information to our clients on whether, so say we had a health department as a client, as Denise mentioned, we were able to give them information so they could take action. Like, I think Denise, jump in here, please, about like distributing masks or doing testing in a certain neighborhood because of data that they had been provided through our program.
Yes, that is correct. Currently, we are no longer analyzing samples in-house for SARS-CoV-2. We are sending the samples out overnight to an external lab to be analyzed. However, we are the specific public health actions, like Kyah just mentioned, there we did see mobile testing deployment get implemented in one of our communities. And we did see resident notification, such as posting flyers, just making the residents aware in that area that there is an uptick in COVID.
For this mobile sampling device, it is quite attractive for the upstream sampling. Do you in contact with the decision makers? Well, what is your contact with the decision makers? You are just trying to reach the local authorities or public. What is your goal with this product?
We are specifically, sorry, would you like to go? Or? We are specifically working with the public health departments and these communities to look at specific targeted areas, like the most vulnerable communities, to then track the viral load over time, to then look at potential upcoming outbreaks. So we are directly in contact with the public health departments and providing the results to them.
Yeah, so oftentimes our client is, potentially a health department, and they're still working to figure out, the best strategies. And so this is kind of another strategy in their toolbox of dealing with a pandemic. And again, looking towards the future, hoping to expand wastewater surveillance, as we talked about, like as cases, hopefully go down and all this, the real power of surveillance comes from also, you have, say, minimal cases, low cases, and then you're surveilling constantly and you get a spike in some small community. Okay, you target that community, you say, let's do mobile testing sites, whatever it may be, and then you can hopefully prevent, this major spread in the future. I think that's sort of where it's headed in terms of the technology. I don't want to say the technology, but I guess the implementation of programs like this. And again, I think that's where our focus is helping, public health departments and helping our clients be able to just use this information to take action to help protect the public.
Do you have any trial in any hub, like airport, a metro station, something like this?
Airports.
I do not believe so.
I don't think so. So we've done, I think, a big focus on vulnerable populations, like schools and congregate care facilities.
Kyah, I think it may be a good solution. I think it is not the case in US, but there are many non-sewered areas in the world, and this can be a good solution for the non-sewered areas. So you can use this mobile device for the for the non-sewered areas. Okay, thank you. Nur, I think we don't have any other question.
Yes, maybe final question I can ask to Till. I'm wondering if it's what he thinks about the importance of having local data. Because the data you're having with the fecal shedding, maybe there are some factors that are affecting, like ethnicity, gender, age. Do you think it's important every country has their own data to be able to modeling purposes?
Good question. I think that in part depends on exactly the issue you raise of how much demographic variability there is. And I think something that we should do in a first stage is try and get diverse data. And if there is evidence that there's a lot of variability, then it would definitely be worthwhile drilling down to get, more specific data that might be more informative for a particular region. And yeah, I think something in light of David's presentation as well, one thing I didn't mention is that, of course, variants might lead to different levels of shedding. So there's definitely a question of how do we prioritize our resources? Should we get more focused on demographics, or should we make sure that we understand vaccination, or do we need to understand variants better? And I think that's an open question.
Before closing, I have one more question to Till. Do you collaborate with the public health people in other countries? Because in wastewater-based epidemiology side, we are in close contact with the other countries. What about the public health part? Are you contacting with the other countries? And yes, for this demographic analysis, it is quite necessary because you need data, okay?
Yeah, so I used to work for the UK Department of Health as a consultant on the analysis of the wastewater data. And it was great to see there that it was quite an integrated response where we had both high-level clinical data, information from the UK Statistical Authority, as well as the wastewater data. So in that sense, yes, I haven't worked with anyone in the US yet because I only arrived a couple of months ago.
Okay, I think we can close. Nur, thanks to all speakers for the great presentations, answering questions, and their valuable contributions on wastewater-based epidemiology. Thank you all for all attending people. You have learned and enjoyed this webinar. And finally, thanks to American Society of Civil Engineers providing us this platform. Have a good day. Let's meet in another webinar. Okay.